I'm going to hit go. I think we can do we can start. You want to introduce Monica?
Okay, sure. Hi, everyone. Thanks for joining us in today's seminar. So Dr. Jonathan Goldenberg is a postdoc from Evolution and Optic of Nanostructure in University of Gent, Belgium, so across the pond. Their lab mainly studying how nanostructures such as fine scales produce not only color, but also provide thermal and antimicrobial function, and how does it evolve by not only comparing the modern specimen, but they also study ancient fossil. So Jonathan is actually my friend from way back. We did master together in Europe about 6 years ago, and he is always crazy about reptiles. Even when he visited me and my husband in Indonesia, the activity that makes him most happy is when we go to see the snake house. He is also an avid photographer, and some of his shots win awards and appear in places such as National Geographic magazine. He really hates when people cannot tell the difference between poisonous and venomous. That is his word. And as a typical Italian, he's really offended by pineapple pizza. So given his passion about reptile, no wonder his research is about reptile too. And today we're gonna hear him talk about that. So Joe, thank you for coming today, and you can start.
Thank you very much for this introduction, Monica. I couldn't agree on all the points, especially the poisonous venomous point and the pizza. So thanks for the great introduction. And yes, thank you very much for this great opportunity. It's a great pleasure being here with you all today. I guess everyone can hear me clear and loud, right? And maybe Monica, just do like this with the thumbs up, just to make sure. Yeah, great, okay. So in today's presentation, we're going to see how the color integument of squamates influence the ecology and evolution of these guys. And before getting to the core of this presentation, I just want to make sure that you have an overview of what you're going to expect in the next 45 minutes or so. So initially, I will provide you some background information to make sure all of us, we are at the same page. And then I will provide some case studies where we're going to see how the evolutionary path of squamates is shaped by their colored integument. And as we're migrating towards the end of this presentation, I will actually assess, possibly even through a discussion together with you, how actually ecogeographical rules are placed nowadays, and if we can actually use them still in terms of climate change and rapid climate evolution. But without further ado, let's get into the topic. Yeah, there we go. I'll also turn on the pointer, and there we go. So coloration. Coloration evolved across the tree of life to perform a plethora of function. Now, unfortunately, I won't have time to discuss all of them today. So what we're going to do, we're going to focus specifically on a very enthusiastic, for me, enthusiastic clade, which is the reptiles, and more specifically, the squamates. Now, color production in squamates is highly conserved, and there are different cells producing colors, collectively known as chromatophores, which are primarily responsible for producing colors in these guys. Now, on the upper layer, we're finding the xanthophores and the iridophores, which generally produce coloration that ranges and spans from yellow to orange to reddish. Then we're finding the iridophores, which are platelets that generally produce whitish coloration. And at the bottom of these dermal chromatophore units, we find actually the melanophores, which are generally producing dark coloration. And the interplay of these actually chromatophores ultimately determines the observed hue. Now, previous studies mostly focused on camouflage, mimicry, social or sexual signaling, but actually coloration have very important thermal properties. And that's because by absorbing and reflecting solar radiation, the color tissue can affect the body temperature. Now, we know that generally bright colors reflect more light and thus absorbs less energy, whereas dark colors in general absorb more energy, which is ultimately transformed into heat. Now, as we've just seen a few slides ago, we've seen that melanin is primarily responsible for the darkening of the integument. And as the sun's energy radiation spans from the UV to the visible light up to the near infrared, it goes without saying that melanin performs a key fundamental role in the thermoregulation of especially ectotherms, because it absorbs not only, as we see here, in the visible spectrum, but also it goes further beyond in the near infrared, which is actually information that many times we are lacking to understand or we are forgetting. Now, ectotherms don't only rely on coloration in terms of heating up. Like, for instance, we know that there are behavioral adaptations that help them to cool down and to regulate better in case of strong solar radiation, but also very peculiar morphological adaptation, as we can see here in the mollusk, that is actually forwarding all the nice droplets with these nice spikes towards this dewlap over here. Now, in terms of sun's energy-rich radiation that reaches in organisms, we commonly assume that all of the energy that reaches an organism comes from the top. But actually, there is a very strong component of heating up an organism, which comes actually from the bottom, from the substrate. And that's because each substrate type has a different specific heat capacity. Now, assuming that not all of us, we are actually accustomed and acquainted with this term, the specific heat capacity is actually the amount of energy needed to change the temperature of 1 kilogram of substance by 1 degree Celsius. But to place it more in layman terms and like with some nice graphical representation, a low specific heat capacity value we can actually translate it into a hot environment, because actually those substrates are more prone to dissipate the heat. So obviously they can cool down, but they can reach much faster, higher temperatures. And that's why we can categorize them as a hotter substrate. Whereas high specific heat capacity values are those that are good to store the heat. And that's because they contain a lot of water, which we know that actually water in a way slows down the heating of a specific substrate, and also doesn't in a way allow to reach very high temperatures. So we can assume actually that the specific heat capacity which goes high, the substrate with the high specific heat capacity, are those that are actually cooler. Now, with all of this in mind and looking actually to this flow of solar energy, we see that there are many different paths that can reach an organism in terms of sun's energy-rich radiation. We have direct radiation, we have reflected radiation, we have the ground thermal radiation, and even thermal conductivity, and so on and so on. But then one of the first questions that dates back to actually the first chapter of my PhD already, I think 4 or 5 years ago, when I started with this project, we wondered if actually there are different evolutionary pressures between the dorsal side of organisms and the ventral side in terms of coloration. Because as we see, there are many different paths and which are exposed to different conditions. And as we know, we said it before, dark coloration absorbs more, whereas bright coloration reflects more. So is it possible that actually the evolution of coloration in different body parts is decoupled? And that's what actually we tried to test, and we tested actually on vipers. And what we found is pretty remarkable. So first of all, we evaluate the ancestral state estimates of color brightness on the ventral side here on the left and the dorsal side here on the right. And what we can see is that, first of all, we found some convergent evolution, where, for example, unrelated genera evolved independently over time bright coloration. But what actually is even more interesting is that if we contrast these evolutionary trajectory representation and estimation of the trait, it does not actually match that of the dorsal side, suggesting directly from a visual representation that the evolution is decoupled between the ventrum and the dorsum in terms of coloration. But as I just mentioned, this is a visual representation. So we actually had to construct statistical model which accounts for the phylogeny in order to understand better if this visualized pattern is in true actually happening. So what we're seeing over here on the upper panel, we're seeing on the y-axis the brightness of the model on the ventral side. And on the bottom one, we see the dorsal values for the brightness. And on the x-axis, we're seeing the different substrate categories, which are spanning from low specific heat capacity to high specific heat capacity. So once again, from hot substrate to cooler substrate. And what we see is that in every single substrate for the ventrum, the coloration is significantly brighter every time, no matter what is the different type of substrate. And this once again gave us the opportunity to infer that the evolution is of the ventral brightness and the dorsal brightness is actually decoupled. But that's not enough, right? Because this is only one side of the picture. What happens if we are introducing other parameters that are influencing the brightness of these species? And that's what we see over here. Now, this is actually a summary from a Bayesian model. But long story short, what we can see over here, we're having the probability of having a bright ventrum or a probability of having a bright dorsum. And we see that for both, the substrate actually is negatively related to the brightness of these body parts, which means that the cooler the substrate gets, the darker actually the integument gets, both on the ventrum and the dorsal side. But as you can see over here, there are actually different factors and different ecological pressures that are shaping the brightness of vipers. Where, for example, we see that the body mass is positively related to brightness for the ventrum, but there is no effect at all for the dorsum. And we see a negative effect for the distribution in terms of latitudinal distribution, where, for example, we see that the higher the latitude, so the further away we're going from the tropic, the darker the ventrum gets. But again, we don't see any of these patterns for the dorsum. But then it flips, where, for instance, we're seeing a negative effect of altitude on brightness of the dorsum and activity time on the dorsum, but no effect on the ventrum. And that ultimately suggested to us that in reality, ventrum and dorsum evolved under contrasting selection pressures. But all of this study actually was based on literature data. So let me explain better. So all the information that we gathered over here derived from literature information. And we know that many times these studies or metadata that they are relying on the literature may actually miss very important information that are out there in nature. So what we did, and that was during my PhD, we went to South Africa, and that was a once in a life, I would say, experience. And we sampled cordylid lizards. So we moved from snakes to lizards, and these cordylids are endemic lizard to Southern Africa. And we selected 12 different species from 23 different sites for an overall of 276 individuals. And we spent almost 8 months in the field and covered an astonishing amount of 16,000 kilometers. Now, maybe this amount of kilometer for American-based researchers, or at least residents from the US, doesn't sound maybe too much, but for European-based, 16,000 is quite a number. So I was very proud to put this in. And the reason we selected those 12 species is because we couldn't obviously go through the entire phylogeny of 58 actually cordylids. So what we did, we specifically selected a representative from each major clade that we have in this amazing lizard group. And there we go, what we find. So once again, we're having on the x-axis the substrate-specific heat capacity from each of the location in which we sample. And we see that on the right side of the axis, we're having the cooler substrate, whereas on the left side, we're having over here the hotter substrate. And what the model actually predicts is that as we're moving towards cooler substrate, it's more likely to find darker integument. But we found actually something very, I would say, intriguing, also a very nice interaction together with body size, specifically the snout vent length of these lizards, where larger organisms are predicted actually to be even darker than actually brighter ones. So there is a very strong effect of size even when it comes to shape the ventral brightness of these lizards. And now, from a macroevolutionary point of view, imagine that you don't want to estimate one state, you don't want to estimate two states, but you want to estimate 3 or even 4 more states all together in one panel. So what we can do is actually to build up a polymorphospace, which is basically a projection of a morphospace in more than basically in PC1 and PC2 axis. So we're reducing actually our system. And then we map actually the ancestral state in this polymorphospace. And then we can see how over time, the different focal species evolved in this area. And if we keep in mind that, for example, over here, the Y, so the PC2 is mostly driven by the ventral reflectance, whereas PC1 is mostly driven by the dorsal reflectance and the body size. So here we go. Now that I introduced everything, focus on the left side of the panel. Now we start actually with the first splits, and it takes very, very long time before the next splits actually start to occur. And what is very interesting over here is that all of these splits started during the Miocene, when actually the Benguela current started to form along the southwestern coast of South Africa, promoting aridity. And that actually completely changed the previously lush and green environment of South Africa. And that likely actually promoted diversification and speciation events, because there were new niches in which all of the different species could actually migrate towards. And all of these diversification events that we see in speciation occurred indeed when we actually see this variation in climates. But as we said before, we only have 12 different species, and the entire phylogeny of Chordalians comprises 68 species, which means that we had to question how reliable are actually our results. And this is actually coming from a very nice suggestion from a reviewer from a previous revision round, and I'm very happy to present you actually these new results that we included in. And basically what we did over here on the right side of things, we're seeing the evolutionary construction of the body mass and dorsal brightness for our 12 study species from field-derived data. Whereas on the left, we're having the evolutionary construction of body mass and dorsal brightness from data collected from the literature. And what we see actually is that overall, the positioning in the space of the focal species that they are marked over here in red, they're very nicely matching the ones that from the field-derived data. And together with other ancestral state estimation analysis and correlation analysis, which provided us a very strong correlation between the two reconstruction, it gave us actually the green light to assess and actually to make inferences in terms of evolution for the chordalids using only these 12 different species. But unfortunately, not everything is bright. Climate change is here, is now, is happening. And that's why I was very curious as well to understand actually how coloration can influence the future activity time of these lizards in South Africa, these chordalid lizards. And that's what exactly we did afterwards then. So we created climate modelings, and in order to do that, we had to collect information in terms of morphology, such as reflectance, or physiology, like critical mean and critical max, and behavioral activities, and that was collected from the literature and our own observation, and macroclimate data, which was then computed in NISHMAP-R, which is a biophysical model, which ultimately provided us with the activity time under different climate scenarios, both in the present, in the future, and to a certain extent in the past. And the first step that we wanted to do was to evaluate actually how specifically coloration, and especially the darkness of the skin can influence the activity time of organisms. And our expectations were that the darker populations and the darker species would be mostly negatively affected by rising temperature, by forecasted rising temperatures. And in order to do that, we specifically focused on one species, the Karoo-geared lizard, because despite being fairly distributed, it also displays remarkable variation in terms of coloration across populations with very different also reflectance values. And you can see it goes from relatively bright to pitch black. And most importantly, I was talking just before about their distribution, and we see here two locations that they're fairly apart, but then we're having some sort of clustering over here. But for the ones that are not very acquainted with South Africa, it's pretty remarkable the microclimate variation that you can find along the southwestern coast of it. And that's something that makes this clustering actually not really an issue, because for example, over here, we may have actually very windy and humid environments, whereas over here we had actually still windy but very dry environment. So that provided us with very nice opportunity to test all these hypotheses for us. And here we go again with our findings. So first of all, let's focus on the left panel over here. On the x-axis, we're having the integument darkness, which goes from 0 to 1, where 1 is actually pitch dark and 0 completely bright. On the y-axis, we're having the estimated hours of activity across the whole year. And we have three different time periods. The present time, represented in gray, the blue line from 2049 to 59, and the 8099, here we go, is the last one over here. I beg your pardon. So what we see is that overall, darker species are predicted initially to be, as you can see over here, the less active. But as we are going forward in time and we're getting towards the end of the century, the darker species actually are the ones that are predicted to go even further up in terms of activity. Whereas we see at the end of the century, on the other hand, the brighter species are actually predicted to go even lower. Now, I'm talking about species, but I should have said actually populations, right? Because we're focusing here on one species. So I'm sorry for that. So we're talking about populations. So the brighter populations are expected to decrease their activity time by the end of the century. And on the right panel, actually, you can see it even more clear, where actually we're having the magnitude of activity change. And if we have as a reference over here the present time, we see that the darker species, again, the darker populations are remarkably increasing their activity time compared to the brighter counterparts. But as you see highlighted over here, these type of models are deterministic, which means we don't have any variance around our estimation of activity times. We have one single result. So we wanted actually to evaluate to which extent these results actually are robust. And one way to do it, at least that's how we approached it, we looked into sensitivity analysis. So that means that we started actually to shuffle the values of reflectance, so basically of the darkness of the skin of the lizards. And we ran actually all these models again to see how it varies all these relationship, how actually the activity time then changes in relation to that. So as you can see, there are two actually panels, one labeled across locations and one within locations. So for the across location, what we mean is that we took the values that are spanning from the minimum reflectance to the maximum reflectance from all populations. And then we randomly started to assign values in between in series and see how actually the model then is predicted to change in terms of activity time. And if we see that we're changing those values with all these different random numbers across locations, we see that all the different models are far different from the predicted ones. As you see, these in dots over here, these round dots are like the present, the 4059 and the 8099 predicted values from our initial data. And in yellow, we're having all the shuffled data and from the shuffled model. Whereas on the right side, we have exactly the same concept, but the reflectance values are all shuffled within populations. So for example, if we're having an individual, which was just out of giving a number now, classified as 0.3, then there was another one that was classified as 0.5 and another one again with a maximum of 0.6. We let actually all these individuals to vary their coloration between the minimum, which was 0.3 and 0.5. And then we did this for all the different individuals within a population. And what we found over here, contrary to the cross-location model, we see that very nicely all the models are all around our predicted values. And that gave us actually the clue that there is a very strong location effect of the coloration of the integument of these lizards, where specifically we see that, for example, is not reported over here, but just to give an example, the darker populations are found in more humid and cold environment, and the ones that are brighter, they're found in much warmer and drier environments. And that was very nice. But once again, that was not enough. I was still actually curious to understand how actually not only one species, but all of these species are going to cope with rising temperatures, with future climatic projections. And that's the next step and the next study that comes in. So first of all, we actually see two panels over here. On the left, we have whatever concerns temperature, on the right, precipitation. On the x-axis, we're always having the variation across the different latitudes. And then we're having all the different locations, the 26 locations that we previously shown you. And then we see that over time, for example, for the temperatures are forecasted actually to increase pretty remarkably over time and also pretty consistently, right? So we see the slope that is going maybe a bit steeper indeed, but is always kind of consistently going higher. Whereas actually for precipitation, for those locations that are found actually closer to the tropics, so the ones with it's inverted over there, but and to make it simple, to the ones that are actually closer to the tropics, they are forecasted how much higher precipitations by the end of the century. But in southern locations, we can see that is getting actually lower than the current precipitation time. So there is quite a shift, a dramatic shift in terms of precipitation patterns. And why I'm showing all of this is because then we wanted to link this information together with our output of the model, where we see that over time, the activity time at a different type across latitude increases. So we see, for example, that lower latitude, so actually at the higher latitudes, the activity time increases. And that could be said as well for the northern, so the more equatorial population and species of cordelid. But we see over here actually a shift where actually by the end of the century, the northern population are actually expected to go lower. And that might be linked actually to the very extreme temperatures that we've seen in the previous graph over here, that temperature may be so scorching and so hot that lizards won't be able anymore to cope with such extreme conditions. But we can also actually visualize this plot in perhaps even a more friendly output. And that's to a 3D plot where we're having latitude over here on this axis, and on this axis over here, we're having the longitude. And on the Z-axis, we're having the activity hours predicted. And the different cones are actually representing the activity time of the different population, of the 26 different populations. So if we start from the past and then we're going to the present, we can already see like a remarkable change over here in the southern populations and to a certain extent in the northern populations. But then even more, if we're going to in the future, in between the 40 and the 59 time, and we're going even further up, we see that the southern populations are giving really a nice boost over here. And by the end of the century, again, this southern populations are stabilizing a bit more. And you can see over here that those are like the few northern populations that they are predicted actually to lower their activity time. So now that we've seen what is happening in the past and what is happening in the future, let's maybe try to summarize all of this, because I expect that I fed you with a lot of information and a lot of data. So it's good maybe to absorb everything. And I think in this way, it's much easier than to go forward for the talk. So in the first study that focused actually on literature data on vipers, we found contrasting pressures for the dorsal and the ventral side. First of all, we found that substrate that they are more cooler and humid, they are selecting overall for darker integuments, whereas substrate that they are hotter and drier, they select for brighter integuments. But then we found also contrasting patterns, where for the dorsal side, we found an altitude pressure for brightness evolution, where higher altitude are expected to select for darker integuments versus lower one for bright, which could be both for UV protection and for thermoregulation. Whereas diurnal species are predicted to be brighter than nocturnal ones, which can be linked to many aspects. But as, for example, vipers are ambush predators, I would expect something like this to be driven by the conspicuousness and by the fact that they have to be cryptic in their environment. Whereas for the ventral side, we found actually a latitudinal effect in shaping the brightness of this body part, where higher latitude selects for darker ventral coloration, whereas more equatorial regions select for brighter ventral integuments. And lastly, for the vipers, we found that actually the bright integuments are selected in larger organisms, which can also potentially be a thermoregulatory pressure because of the thermal inertia of larger organisms. So maybe just to explain you this concept, so we know that actually organisms that are larger, they have a larger thermal inertia, which means they need more time actually to cool down or to heat up. And because potentially vipers are ambush predators, so they don't move a lot across the landscape, but they stand still, it's very important for large organisms to maintain their body temperature as better as possible. And that's possibly one explanation why brighter integuments are selected in larger organisms. But then from the field data and using cordelid lizards, we found actually exactly the same patterns for the substrate specific heat capacity. Once again, where dark integuments are selected in a more humid and cool substrate, and bright integuments are selected in hot and drier substrate conditions. And for the ventral side, we again found actually this exact same evolutionary pressure, where larger species are predicted to be brighter than a smaller one on the ventral side. But then we took the step and looked actually how coloration influences the activity time. And so actually our proxy of fitness in the future of the cordelid lizards in South Africa. And we found actually that the activity time of darker populations is expected to increase by the end of the century, much more substantially than the brighter ones. And then overall, if we took actually an holistic approach with all the 12 species, we found actually that, yes, populations and species are expected to increase overall their activity time. They are expected to do so. But by the end of the century, some northern populations are actually expected to decrease this activity time, which is possibly related to the scorching temperatures forecasted by the end, again, of this century. But given all this, now we really focused on few clades, right? We looked into vipers and we looked into cordylids lizards. But then, like, my question was, can we actually find some global trends? Can we actually try to infer how color brightness evolved throughout the squamates? Can we find global patterns that are shaping the coloration of these organisms? And that's what we did next. And that's something actually pretty new that we're about to submit. So it's a recent work from our lab in collaboration with Groningen University. And what you see over here is the distribution of brightness. Well, I beg your pardon, I forgot about my clock now. And yes, okay, great. So we see over here the distribution of brightness across 1,235 species that we selected from phylogeny. So I won't get too boring and try to explain you all the details, but the main point over here is that we tried actually to see how, first of all, brightness is distributed across the globe in squamates on the dorsal side, specifically talking. And what we can see is actually that there are some clustering, for instance, over here in all the Saharan and Sub-Saharan region, we can see some more brighter integuments, whereas if we look here in more the tropical regions and rainforest region, we see more dark integuments, and same goes over here in the Amazonia, where here, for instance, in the States, we see brighter integuments. So we questioned and we hypothesized actually, the first question is like, where do we find bright or where do we find dark colors? And then that led us to actually hypothesize that bright integuments will be selected in open areas such as grassland, prairies, and so on, whereas dark integuments will be selected in closed canopies such as rainforest or the like that. And this was not simple, right? Because obviously we had to classify a lot of information across all of these species, but this was eventually possible. And also we did not only include this specific variable of the vegetation structure, but we accounted actually for other ecological environmental information that most likely influence the brightness of squamates. And what we did is actually, first of all, to plot, so visualize actually the ancestral state estimation of brightness across all our different species, and at the tips to contrast it with the vegetation structure that we classified as closed, fossorial, open, and open-closed. And what we see is, for sure, we found some clustering over here, where, for instance, dark integuments over here in front fang snakes mostly relate possibly to open, closed, and more closed environments. And that's the opposite can be said for the lacertids, which are much brighter, and we can see they're mostly selected in open areas. But once again, what we're seeing over here is a visual representation of an ancestral state estimation. It's not actually a statistical analysis. So what we had to do is, again, to perform some phylogenetic models to understand if the patterns that they are visualized over here are actually occurring or is it just phylogenetic signal. So, as we mentioned before, we actually collected not only vegetation type, but also other ecological parameters that most likely influence the brightness of species, and namely the altitude, the conspicuousness, the activity time, so the circadian rhythm, as we said before, obviously the vegetation type, the body mass, and the latitudinal distribution. Now, I omit over here for simplification all the different interactions, because actually what we're going to see over here is already much more than complex, and to digest everything would have been too much, but it's very straightforward how it's going to see over here. So bear with me. So initially, we ran all of these models simultaneously at global level, so including the entire phylogeny. And what we found, there are very interesting patterns where, for example, the altitude, the higher the altitude, the darker the integument is predicted. And then the more conspicuous species, the brighter. Nocturnal species are more likely to be darker, and vegetation type, the more open areas are more likely to be bright, where closed areas are more selected for darker integuments. Negative relationship between the body size and the integument, and again a negative relationship between latitudinal distribution and body size, and brightness. But then we know that the phylogeny, so the input data, can be very influential when it comes to define the output of a model. And we know even more that every clade actually within this actually big global model within the squamates is subject to contrasting evolutionary and ecological pressures. So to this end, actually, we started actually to build up model for different focal families. And what we see is actually contrasting actually patterns, right? We can see, for example, that altitude is not consistently here, conspicuousness again, and so on and so on and so on. But I guess you already see it. Vegetation is consistently ranking as a very important parameter in shaping the brightness of all these different clades. And it's always in one direction, where actually open habitats are selecting for brighter integument, whereas dark, closed canopies are selecting for darker integuments. And this was very remarkable because, to be honest, I was not really expecting to find such a consistency, such a strong pattern across the entire phylogeny and even within all the different clades. And also quite interesting, I would say, is about the body mass. We found actually some sort of strong influence in shaping the brightness evolution in 3 different clades. And these 3 different clades actually occur to be one of the smallest one in terms of body size. And our inference is that smaller organisms, in order to heat up, if they have for sure a larger or a smaller body, the coloration is extremely important. So at the moment that take any given small subject and you change it in color, like a darker color, most likely it will heat up much faster than a brighter counterpart. And that's why we are suspecting that this trend that we see in body size is significant for those clades that they are displaying relatively small body sizes. But now that we found this very strong pattern for the vegetation structure, we wanted to understand actually how the evolution of brightness occurred over time, specifically how the rate of evolution of brightness changed over time in relation to the vegetation structure. And one way to do that was actually to retrieve information about past climatic conditions. So it's very hard to find what was actually the vegetation structure overall in the globe in a very specific location. But what we can know is actually the average temperature at global level through the ratio of the oxygen 18 isotope and the oxygen 16 isotope. And to make it in simple terms, lower values of this ratio indicates relatively warmer environments, and the higher the value goes, the cooler the environment gets in any different given time. And if you look over here on the left side, so panel A and C, we can actually see the evolution rates of the different focal clades that we've seen in the previous table. And we see that overall, over time, it seems to be pretty stable, right? The evolution rate, so the speed of change of brightness over time. But it started actually to remarkably increasing over here at this time point between the Miocene and the Pliocene. And that's where we're getting now to the second two plot, B and D, because here we're having a zoomed in version of the previous plot. And what we see indeed is that at the interface between the Miocene and the Pliocene, and more afterwards between the Pliocene and the Pleistocene, we see very strong changes in terms of increasing of brightness evolution rate. And this, we speculate, might be related to the remarkably changing of climate, like it got much, much cooler compared to the past. First of all, with the Pliocene climate shift that occurred roughly around 5.3 million years ago, followed by the Quaternary glaciation 2.5 million years ago. And that was actually a very interesting pattern, because then we actually could relate and we actually can infer that the habitat type we change due to this remarkable change in the temperature ultimately influence also the adaptation of these species into new environments. So to summarize all this last point, we've seen that the vegetation structure has a very important force in shaping the brightness of squamates overall, and is consistent, where close environments are selecting for dark and bright ones are selected in open habitats. Body mass, on the other hand, seems to be only influential for smaller, for clades that they are displaying relatively small body sizes. And as you can see, for the largest one, we didn't find any patterns. And the most interesting part, at least for me actually, was even to not find any pattern of latitudinal distribution effects on the brightness of these species. Now, if you remember on the vipers, we found such trend, but only on the ventral side, not on the dorsal side. And that's the reason actually that then I was very surprised, because I wanted to understand why actually we don't find any pattern like this. And that brings us to the last topic, which is the ecogeographical rules. Now, I want to link this directly because it was the Berger rule that I was expected actually to find, right? Like, and specifically that larger organisms are most likely to be found in higher latitudes in order to maintain their thermal core more stable in colder environments. But what is the story about here is that these ecogeographical rules were actually developed in the last 150 years or so. And since a much smarter person than me for sure started to postulate these different trends of phenotypic change across the biogeographic perspective, they were developed 150 years ago. It's a huge difference from nowadays conditions. So can we actually still talk about Allen's rule and Bergman's rule and find actually these same patterns nowadays? This came out actually as a relatively recent forum paper that we wrote. And the take-home message from here is that also I'm very happy to discuss further about this during the questions, if we have time afterwards, is actually, can we still use these ecogeographical rules as traditionally interpreted? So let me be more clear. For example, in a case where we are talking about a rule is meant to be broken? Possibly yes, but then is it a rule? No, it's a trend. So the first point that actually we made over here in terms of precautions, if someone wants to use ecogeographical rules as a study, is to, first of all, use the word trend instead of rule, because most of the times when we're trying to find patterns across time and space, there is always an exception and it can't be a rule anymore. So that was one of our precautions, to say, okay, we want to still use ecogeographical rules, fine, but let's call them trend instead of rule. The second point was actually in terms of what's the benefit of testing for this rule. Like, we've seen that these ecogeographical rules are very controversial, and there are constantly pros and cons, and like constantly studies that they are supporting and not supporting. So what's the reason actually to start a study with an hypothesis that is testing for an ecogeographical rule? Does it make more sense to test actually a mechanism which is behind maybe an ecogeographical rule, but why to label it from the beginning and categorize it? So actually, we found actually much more interesting to discuss this from a perspective of a mechanistic point of view. And then as they were developed 150 years ago, they started to be developed 150 years ago, and many translations went around, they changed actually the original postulation. And so what actually is the ecogeographical rule that we are referring to? And specifically, there is one, the Glogger rule, which is very much debated, because many times we are assuming and calling it in many different studies as, for example, that more humid environment are selecting for darker integuments, but actually is a combination of being humid and hot, which then there is all the mechanistic insight of, for example, camouflage or antimicrobial protection and so on that comes into play. So again, these states, we have to be very careful when we are naming even ecogeographical rules, because by the time that it was actually emancipated and by the time that we're calling it now, there are different versions of it. So we have to be very careful on what actually we are calling. And now we come actually to the fourth point, which is actually climate change. So these rules were developed 150 years ago, and still till now, we are using them. But the climate really remarkably changed, and there are many different patterns. So don't you think maybe they are a bit outdated, some of them? Do we really have to test for them as they were traditionally interpreted? And the last part and the last point is if the study organisms occupy geographical or environmental gradient. So the ecogeographical rules were actually emancipated once again for geographical gradients, not really environmental gradients, but we are using them nowadays as environmental gradients as well. But then they're deviating from the initial description of the rule. So again, does it make actually sense to use the classification of an ecogeographical rule to define a mechanistic pressure, such as an environmental gradient pressure? With this, I would like to thank you all for your attention. And despite the initial technical difficulties, I hope you were able to follow me. And I'm very grateful. And I want to thank again Monica for inviting me to give this talk and to share the findings with you all. And I would like also to extend my gratitude to my lab, to the EON group, and to all its members for the continued support and for all the help and constant push that I got for all of this work. And I would like actually to conclude with a final remark, which is quoting physicist Brian Greene, which actually is said once that skepticism is an art, not an algorithm. Whenever we approach new research perspectives, we may inadvertently cut off ideas that we're simply not able to process in this moment of time due to lack of foundation. And that is one of the many reasons that we have to keep our eyes open and look for as much biodiversity as we can, as we never know where the next bio-inspired discovery will come from. Thank you very much for being with me.
All right, thank you, Jonathan. So probably if anyone have a question, they can set it out now. Either like in the chat or just like directly.
I have a question.
Yes, Rebecca.
Great talk. Do you have any intuition on how the food availability might scale for the populations in South Africa that are the model predicts that their activity levels will go up with changing climate?
That's a very good question, because we also pointed out in the manuscript actually that the activity time for us was really a proxy which had many limitations, and one of them was obviously the no information about food availability, right? So we clearly pointed out that that was a lack of information that we could not infer, as well as the interaction between and across species and prey predation interaction. But if I were to make a wild guess, I would say that everything depends on their insects, right? So basically on the preys of these chordalids that they're mostly preying on. And if I were to think, so far the southern populations, so the ones that are expected to increase much more their activity time, I would expect actually that the insect as well and their preys then would also increase, because there are also ectotherms, and also they are therefore I would expect them as well to be even more available to a certain extent. Obviously, we have to consider as well for the insect as well their interaction with the environment in terms of plants. And we've seen, for instance, that precipitation is going to decrease in the south. So that might be actually a counter effect to the fact that the temperatures are going higher, but there is less maybe precipitation forecasted in the far future in the south. And that might actually inhibit the population growth, or in general the insect growth in the southern part of South Africa. And then we can make similar estimations for the north. But again, like all of this is a very big wild guess, because for sure there is going to be an effect of prey availability, obviously predators as well. So it's very hard to make a consistent, I would say, estimation for it. But it's definitely something that has to be accounted if we want to make, for instance, species distribution ranges changes and so on.
Great, thank you.
Sure.
I have a question.
Yes, Kristen.
Like turtles, are their egg sex determined by temperature?
I don't want to say anything wrong, especially on the vipers, but for chordalids, if I'm not mistaken, yes. And that's also one of the points in our biophysical models. We didn't include the dynamic energy budget information in there. So we, in a way, in NISHMAP-R, in the specific package that we use, we suppressed actually that option where it doesn't look into the generation time of offspring and so on. So for sure, it's very important as well when it comes to account for changes in terms of activity time. But also there is another parameter that we pointed out in our manuscript, is that also the adaptation, so the phenotypic adaptation, and how fast actually lizards, but any other organism can change coloration in response to changing climates. So our study was really in a way limited to the fact of understanding how the present day coloration of lizards would change the activity time over time. And and that was basically what we did. So we didn't try to give actually again species distribution models information. We really wanted to see how coloration can influence the activity time. That was the goal of the project.
Thank you.
Thank you.
I have
Yes,
a question. Maybe you talked about this, but do you have a guess how much of the association between darkness and, like, forest cover versus openness is due to camouflage versus, needing to absorb sunlight sort of things?
Yes.
Thermal
So actually, we accounted for it in our models, right? So we didn't have only vegetation structure, we also had conspicuousness over there. So we actually had an entry in our models that was literally a binary code. So if a species was classified as cryptic or conspicuous, or as well also uncertain. And that actually, in a way, follows a previous classification system that was used in frogs, and also most recently, I forgot the study system, but it came out from the Stewart Fox lab pretty recently. And they also used that classification system. But there is a huge but. This is actually a classification that works on the visible spectrum. So basically, we as an observer, we are watching an image, or like we are assessing a specific individual, and we see how it blends with the surrounding environment. But as we've seen, the visible range and the human visible range is only a small fraction, actually, of what there is out there in terms of light. And we know actually that the organisms, especially lizards and snakes and so on, they have completely different visual system than ours. And that might also be explained, that also might explain why we don't find any consistent pattern of conspicuousness. Because, for example, my wild guess again would be that there is quite a strong effect with the visible spectrum of birds, where, for example, birds many times are the predators of these guys, in general, of many squamates. I'm not talking now about the Komodo dragon, but most, let's say, squamates around there are like in the range size for many birds. And then maybe there is some sort of interaction there between the visible spectrum of the birds and the color integument of lizards, or in generally squamates, and possibly an interaction with the environment itself.
Maybe we should thank Jonathan again, given the time.
Thank you.
Are you what's happening now, Monica? I forget.
I don't think we have a tea time because no one registered.